Great. I think we can start. Happy 1st of April, everybody. I hope you're having a good time. A little bit cold here, but the spring seems to be picking up. And today we have another UAC seminar where we're going to hear from two of the group leaders that are having projects here at the Uppsala Antibiotic Center. And we are going to start today with Sanna Koskiniemi, and we'll have a break and then continue with Olof Lindahl. Like always, you can either write your questions in the chat and I will voice them out to the speakers during the Q&A part, or you can also open your mic and pose the questions yourself. Just have in mind that the meeting is being recorded. With that, I would like to introduce our first speaker for today, Sanna Koskiniemi. She's been with us from the very beginning of the center, and she's going to talk to us about her project looking into targeted eradication of bacteria. How can we actually have a way to really target the bacteria we want to eliminate for the cases that we want to eliminate them? She's working with PhD student Petra Virtanen, and hopefully they will actually have more papers out soon. They have a couple of them already, and it's my pleasure to give the floor to you, Sanna, and see how the project is going and see some of the updates. Thank you so much.
Thanks for the really nice introduction. Now I'll just see if I can figure out the screen sharing.
I believe it's better if you stop sharing, first open it in full screen in your computer and then go back to the Zoom meeting and then share that one. That will work.
Because this doesn't work?
I mean, it works like this. Yeah. It's just that now it's not on presenter mode, right?
What does that mean?
Do you have a PDF or a...
it's a PowerPoint, so I'm not sure what you're seeing. You're not seeing just my slides?
I'm seeing just your slide, but it says, like, for example, like there are some words that need to be fixed and stuff like that because it's not on presenter mode, I believe. Not so sure, but we can see it, yes.
I can stop.
I think if you put in presenter view first and then go back to Zoom and share screen of that full screen application, if it works.
I can't do that. Sorry,
okay,
it doesn't
no
work.
problem.
I can't go to Zoom if I do that. This is how I gave all my lectures.
All right, yeah, it works.
Okay, we'll do it like this. Sorry if there's some words that you can't see or whatever you were saying, Eva. Thanks for having me. It'll be fun to talk about this project. But although who I think should be talking about it is probably Petra, because she's the one who's doing all the hard work. But I'm also gonna tie in at the end to some other projects that we're doing in the lab that are not exactly the UAC project, but still very related. And I think it could be interesting for the listeners to hear about this. So targeted eradication of pathogenic bacteria or any bacteria. The talk I'm going to try to give you an idea of why. Why do we want to do this? What's the purpose? How are we doing it? And which bacteria can we inhibit? And then I'm going to give some indications whether this will actually work or not. But starting out with the why. So why do we want to eradicate different pathogenic bacteria? Well, I think we're all very well aware current situation in the world that bacterial or infections are a major killer of humans still, even though we are in the 20th century. But of these infectious diseases, this is pre-COVID, we have about 15 million deaths by infectious diseases annually. And of these, some are bacterial infections, some are parasitic infections, and many are, of course, viral infections. So our interest is in the bacterial infections, which are, of course, a part of them are lower respiratory infections, but there's also a big part, which is diarrheal diseases and tuberculosis. So our main focus are on infections like diarrheal diseases and also urinary tract infections. Even for just diarrheal diseases, although things are improving every year that goes goes, it's still a major killer in the world. And the people who are affected are mainly children under 5 and old people over 70, as for most infectious diseases. And the people that are affected are not here, not in the Western world, but mainly in the developing world, in Africa and Asia. So we think that by finding means to eradicate specific bacteria, pathogenic bacteria, we could perhaps develop means of preventing infections, because for many of these diarrheal infections, you don't use antibiotic as treatment. It might not even make the infection go away. It might worsen it in many cases, and it is self-limiting. But the reason that people still die is because they're young and can't handle their fluid loss. But if we could somehow prevent these infections from occurring, that could save lives, we think. In addition to this, we have, of course, the problem that you're all very well aware of, since this is a seminar from the Uppsala Antibiotic Center, and that's that we have an increased problem with antibiotic resistance. And this is just some late figures from showing deaths associated with resistance or attributable to resistance, showing that we have many people already in Europe dying from resistant bacteria. E. coli, Klebsiella pneumonia are typical causative agents of urinary tract infections, as well as diarrhea for E. coli. And I will mention these strains a little bit later as well. And this, of course, doesn't affect us as much in Sweden yet, but when we travel to regions where prevalence of resistance is higher, we have the increased risk of taking these resistant isolates and bringing them back home. And this will then increase the prevalence of resistance also here in the Nordic countries, where we're still very lucky here, just showing the prevalence of carbapenem-resistant Klebsiella isolates in Europe. But just by the little vacation down to Italy, we will increase the transmission of resistant isolates. So we think if we can specifically eradicate or target specific bacteria, we could not just prevent or cure infections, we could perhaps also cure these carriers of multi-drug resistant bacteria returning to Sweden, or in any way prevent transmission. Maybe we could even prevent them from ever becoming a carrier of multi-drug resistant bacteria. Because for every bacteria that wants to infect us or colonize us, they do need to get into the body and take care of the normal flora, which is already there. So in order to be able to cause an infection or spread resistance, you do have to colonize a host. And the normal flora is there already in our skin, our mouth, and in majority of it is in our intestines. We have about 2 kilos of bacteria in our intestines. And these bacteria, they help us, they provide us with vitamins, and they also function as an invisible armor that protects us against infections and pathogenic bacteria, as well as from a multi-drug resistant bacteria. But so if this is in place, how come we ever get sick? Well, sometimes you can have a breach in your armor, of course, but you could also be that some bacteria are just better at colonizing an intact niche. So even though the normal flora is there, they might be able to outcompete that normal flora and take their space. And this is what we're trying to understand in this project. How can some bacteria colonize an intact niche? And can we use that information to prevent that from happening? Can we make superbugs, super normal flora that just protect us against infections, that protect us against incoming multi-drug resistant bacteria? So then we have moving into the how. So how do bacteria colonize an intact niche, and what do they use to take out other bacteria? Well, antibiotics is one example. Most antibiotics are bacterial products produced by bacteria to fight off other bacteria. We're not so much looking into that. Instead, we are studying the other things that bacteria do to take out other bacteria. And that includes secreted bacterial toxins known as bacteriocins, as well as toxins, small protein molecules that are delivered by contact-dependent mechanisms. And today I'm just going to give one example. I'm going to focus on the contact-dependent growth inhibition system. But just first to give you an idea, what do I mean with toxin delivery in the first place? So what I mean is that some bacteria harbor systems where they express a toxin. It can look like this. It can be a stick on the cell surface. And when they meet another bacteria, they deliver a toxin part of that into that bacteria. And the toxin goes in and it arrests growth of that receiving cell because the toxin will degrade essential molecules of that cell. For example, DNA, RNA, or kill its source of energy, the membrane potential. Of course, these systems, they come with security for their own kind. So they produce another protein, which we call an antitoxin, and it kind of binds its specific toxin and it blocks its activity. So basically, in this case, the Pac-Man can't go in and degrade the DNA anymore because there's another protein sitting there in the active site. And this allows the bacteria of their own kind to continue to replicate happily ever after. So these delivery systems, as I mentioned, they come in many different flavors, although the toxins are rather similar. So they can be secreted and just taken up by neighboring cells. They can be delivered by this big spear, which is a cell puncturing device that bacteria use to deliver a cocktail of effectors into here. Or they can be present on these sticks on the cell surface, which we call contact-dependent growth inhibition. And that's the systems I'm going to tell you a little bit more about now to understand how we could utilize these systems to arm probiotic bacteria to protect us against incoming pathogens and multi-drug resistant bacteria. Because we need to understand how the systems work. Who can you outcompete with what system? And how does the toxin delivery work? What are the limitations in order to be able to use these systems? So zooming in on contact-dependent growth inhibition as an example, this system is encoded by 3 genes. You have a B gene that makes an outer membrane transporter that transports this large protein, CDIA, to the cell surface, and it looks really like a stick on the cell surface. And at this end, it has a little domain which encodes the actual toxic activity, the Pac-Man, remember? And when the cell, so here's just a picture showing that these are really sticks on the cell surface. You can even see them here in this electron microscopy picture, small sticks on the surface of a mini cell in this case. And when the cell comes in contact with a target cell that has the right protein on the cell surface, this stick can bind to that protein. And when it does that, it undergoes a proteolytic cleavage event. And this frees up the toxin here, allowing it, where did that slide go? Sorry. Allowing that to go into the target cell and either become a Pac-Man inside the cytosol, cleaving DNA or RNA, or it can insert itself into the membrane and kill the membrane potential that I mentioned. So, looking at this, where do we have limitations to when the systems will work and when they will not work? It is mainly in the interaction between the stick and this protein on the cell surface that we find limitations. So different molecules of CDIA, the stick, they look very similar for the main part. So everything that's white here is identical between these 3 classes of CDIA molecules. But then you can see that there's more color in certain regions like here and also at the end. And this end here, that's the toxin part. So that's why they're different. They have different toxic activities. But also this region varies. And the reason it does so is that that's what directs the binding to the receptor. And early work done by Petra showed that for class 2 molecules, this region was not, it's rather specific in which bacteria it will bind to, which proteins on the surface it will bind to. It will bind to OMP-C and OMP-F, but it's not strictly species specific. So what she could see by investigating this, by cloning the OMP-C protein from many different species into E. coli, she could find out that bacteria expressing class 2 systems could actually inhibit E. coli cells that express the receptor from E. coli or Salmonella or even Enterobacter cloacae. And what you're seeing here is a competitive index. I'm not going to dwell on what that is, but just tell you that it's a measure of how the ratio, so you take 2 bacteria and you mix them at a ratio, and then you look at how does that ratio change over time. And if the number becomes bigger than 1, it means that whoever is the inhibitor is winning. And if the number becomes smaller than 1, it means that the target is winning. And here you can see that a target that doesn't have the cognate receptor, it actually wins over the inhibitor. Whereas if it had any of these receptors, the inhibitors were winning. So she also repeated this with the actual bacteria, and she could see that this bacteria with the class 2 CDI system could inhibit indeed E. coli, Enterobacter, Klebsiella, as well as Salmonella. So this is a kind of a broad spectrum inhibitory mechanism that allows you to inhibit Enterobacteriaceae of different species. And looking at the WHO critical pathogens list, where we really need new antibiotics, we find some of these bacteria that our system could inhibit on this list, including Enterobacteriaceae like E. coli and Klebsiella, Salmonella, and Shigella, which is mainly E. coli. Okay, so very good. We have some system. We know it works against the pathogens we want to target. So can this work in real situations? So then we move to looking at these systems in a host, and we use mice. And typically what we do is that we use a mice model where we treat the mice with antibiotics to disrupt that normal flora just a little bit. And this then allows us to colonize the mice with any bacteria that we like. So in this case, we colonize them with our laboratory E. coli strain. So now we have a normal flora where we know which Enterobacteriaceae we have in there. We have one that we know what it is. Then we feed the mice either another laboratory strain, which has a different antibiotic marker, so we can separate it from this one. You see it's blue instead of gray. Or a laboratory strain that is carrying one of these CDI toxins that I was just describing to you. And then we just collect poo from the mice and we plate on different antibiotic plates, and we try to find out, is any one of these better at colonizing a mice than the other? And what we found was that the E. coli, which had a CDI system, they were better at staying in the mouse. They still went down during the first few days, but they were better at staying there than the E. coli strain that lacked the CDI system. But this is still not very efficient colonization. So we wanted to find something, a more clinical strain that was better at colonizing than our laboratory E. coli. So then we got our hands on a strain that comes from the Charles River laboratory. It's called E. coli R12, and it was causing recurrent urinary tract infections in the animal house at Charles River. So they had a problem with the strain because it just kept spreading. So it seemed to be a very good colonizer. So we did that same experiment that I just described, but now we're using either the lab strain or this R12 strain. And now what we find is that the lab strain is very bad at competing against this super colonizer within the mouse. So the super colonizer comes in and it stays rather stable, and it completely outcompetes the E. coli lab strain in these mice, whereas the lab strain was not able to do this to the same extent. Actually, if we just took mice and never treated them with any antibiotics and we feed them R12, we could see that R12 was actually able to colonize these mice as well. So even though the numbers first went down, after a little while, they came back up, showing that R12 is a very good colonizer. So now you're wondering, what does this R12 have that makes it such a good colonizer? Well, it turns out it has a bunch of colicins. So it has 3 colicins, as well as 2 of these CDI systems. So colicins are those secreted bacterial effectors, okay? So of course, we want to know if they're active. So we knocked out the CDI systems in R12, and we could see that a wild type then could again outcompete these strains, but a knockout that didn't have the toxin could not. And similarly, wanted to find out if the bacteriocins in this strain were effective. Remember, they're secreted. So we do this a bit differently. We take the supernatant and we filter it, and then we add it to growing cells and see if it inhibits growth. And we found that it does. It inhibits growth very effectively. So wild type supernatant, which is able to deliver all colicin, is very good against all these strains, except that one. And the reason for that it's not so good for that one is because it lacks the receptor for 2 of the colicins. So then we did a number of combinations to figure out which of the colicins are actually effective and which are not. And we came to the conclusion that colicin B and colicin Y are both contributing to this very efficient killing by R12 of the laboratory E. coli. So then we knocked out all these colicins. So here's a strain lacking all the colicins, or we knocked out both CDI system and 1 colicin. And at this point, we didn't have the knockout that lacks all 5 systems. We do now, but we haven't had time to test it yet. And then we repeated our experiment in mice where we colonized the mice first with the laboratory E. coli and then feed them R12 or the mutants. And what we found then is that the mutants were not as good as the wild type in getting rid of the MG6055 laboratory normal flora, although they're still better at colonizing than the laboratory strain. So they're still staying very stable. So we haven't taken away all their ability to colonize, but they're not doing as well, we think. So it will be very exciting to see what the quintuple knockout will look like when we have a chance to test that. Okay, so it seems like competition systems do play a role in colonizing a mouse. But what about humans? I mean, we're different, right, than mice? So now I'm gonna just tell you, with the last few minutes I have left, a little bit of another project that we are doing in collaboration with the University of Bergen. And this is work done by another PhD student in my group, Donna Lee, and Oda, who is a PhD student in Bergen. And here we're using, so in Bergen, they have a very cool model to investigate ETEC infections, where they use a human challenge model, where they actually feed med students quite a lot of a pathogenic ETEC, and they analyze the stool samples and serum of these infected individuals. Now you're thinking, how can this be ethically okay to feed pathogenic strains to humans? Well, it can be okay because there is a very big need for an ETEC vaccine. And the reason they're setting up this trial is to try to find means to prevent a lot of child death. Because ETEC causes about 220 million cases annually. 75 million of those are in children under 5. And we have almost up to 40,000 deaths in those children every year. So it is well motivated to have a human model to study ETEC because there are no mice models available. So what they find in these challenges is that when they challenge individuals. For one of these strains, first of all, they had to use a very high dose to get any of the individuals actually getting sick. Whereas for other ETEC isolates, you need much less ETEC to be fed to these individuals. And also, some of the individuals, even the ones receiving a very high dose, did not develop any symptoms. So when I heard about this, I thought it would be very interesting to look at the normal flora of these individuals prior to the challenge to see, is there anything in this normal flora that could perhaps explain why they are not getting sick? So we zoomed in on the ones that got the highest challenge to begin with, and Dana set up this high throughput competition, because we're talking about 700 isolates that we need to screen, where she basically labels the ETEC strain fluorescent and the normal flora is not fluorescent. And then she mixes the cells on agar spots in a 96 well plate, looking something like this, lets them grow, and then she measures the ratio in a flow cytometer instead of doing a manual counting. This doesn't give us the whole picture about the competitive indices of these bacteria, but it does show that some of the isolates here, just showing a small subset, and this time, if you're inhibiting, if the normal flora is inhibiting, the numbers are going towards 0, whereas if ETEC is winning, the numbers are going up. And what we can see is that there's quite a big difference between different isolates. So some are very good at inhibiting down here, whereas others are not as good as all. And if we look at some of these isolates more in specific, we can actually find that they're pretty good at inhibiting many pathogenic strains, including uropathogenic E. coli, enterotoxic E. coli, salmonella, as well as other E. coli strains. That's what we've had time to test so far. And how they do it, we don't know. We haven't gotten the genomes of these isolates yet, but we know it's not a colicin because we can't see any activity from the supernatant like we did with R12. So it seems like all these individuals that were challenged with ETEC, and they needed quite a lot of ETEC to become sick. They actually had very good normal flora that were able to outcompete that ETEC strain. And perhaps that could be an explanation to why they needed to use so high doses of ETEC to begin with. But this is something we will have to find out as the project moves along. And I think my time is pretty much up, but I just want to mention that making a super colonizer, we also have to ensure that it doesn't become pathogenic. And we intend to do that both by doing biocontainment, so basically making it auxotroph or something, so it cannot grow except when we want it to. But we also going to arm it with a CRISPR system that protects it against horizontally incoming genes. So it can't pick up virulence genes, and it cannot pick up the genes that made it auxotrophic to begin with. And we've done some work on this where we basically built a CRISPR system that we can move around and that now protects against a bunch of multi-drug or drug resistance genes, so antibiotic resistance genes. And Dana did some testing of this system recently by growing cells that have the system or don't have the system and just transforming in plasmids with different antibiotic markers on them. And what we see is that if they have a target, so if they can actually inhibit transformation of or cut this particular gene, there is no transformation in the strain that is CRISPR plus. These are colors are wrong, I realize. So if you have the correct spacer, you cannot take in CTXM15, but you can take in TEM1, which gives ampicillin resistance, since you don't have a spacer towards that. And with that, I'm just going to conclude and say that, yes, I think we know that contact-dependent growth inhibition systems can function cross-species against different Enterobacteriaceae. We think that we can use CRISPR to protect against incoming virulence or metabolism genes. And we have shown that ARN bacteria are better at colonizing an intact niche at least if the bacteria in the knees are susceptible to those toxins. And with that, just thanking the people involved. So Petra, of course, and Dana. We also have Evelina, who did a lot of the mice work, and Lisa and Marlene, who did some of the pioneering work in mice. And then, of course, all the people in Bergen, as well as Uppsala Antibiotic Center and the Foundation to Prevent Antibiotic Resistance for funding. And all of you for listening. Thank you.
Thank you so much, Sanna. We have a couple of minutes for questions. If you have anything, you can raise your hand or write it. Meanwhile, I have a question, see if I understood correctly. So with the colicins, you can do it with the supernatant test. That means that the contact between bacteria to bacteria is not really needed. It's just that excretion is present in the environment where the other bacterial strain is going to be. Is that correct?
Yes and no. So in the case with laboratory coli, which have short LPS, we find that you do not need to be in close proximity when delivering colicins. Then it's enough to get it from the supernatant. When trying to inhibit a strain that has full-length LPS, however, we only see effects if we actually let the cells grow together on an agar plate, suggesting that perhaps then you need to come in close proximity of the cell that you're inhibiting.
Mm-hmm.
Did that make sense?
Yes, it does make sense. I was just thinking that if it's the supernatant, if it's this molecule in the supernatant, you could think about treatment that will prevent colonization. For example, when you are going to be exposed in high-risk areas or something like this, you could think about a pill that will have just that colicin and might work to prevent the colonization by these strains. But I guess you can do the same thing with a pill that contains the inhibiting strain as well, right? That's the idea.
That's the idea, yes. And that's why it needs to be biocontained, so we don't spread it all around the world afterwards.
How do the probiotics work? It's just the bacteria that are dry in a pill, and then when they come into the intestinal tract, they become alive and they start dividing?
So that's one way of doing it. But I mean, I think the best example of a live strain that we all drink is Dukoral, which gives you some protection against ETEX or traveler's diarrhea, as well as other bacteria. That's a live E. coli that you get from the pharmacy, and it's in a little vial, and then you mix it with samarin, so bicarbonatic acid it is, I think, and you just drink that before, like a week before you go to Egypt or whatever. I don't know if anyone here has also had Dukoral at some point.
Interesting.
So it doesn't have to be in a pill as long as it can survive the way down through the stomach and into the intestine.
I didn't really maybe understood from the test, but does the inhibiting strain needs to be on the intestinal tract for some time before it gets exposed to the colonizer strain, or can they come both at the same time and will it still work?
So the experiments we have done are mainly to first have the normal flora present and then challenge with an incoming pathogen, because that's how we think it works in the normal situation. I mean, you have a normal flora there, and in order for you to be good at colonizing, you need to be able to disrupt the intact flora. If you would send in them at the same time, the effect is bigger. So then it's much easier for them to outcompete the sensitive strain.
I understand.
And in terms of R12, we couldn't even do that, because as soon as we mix them, the supernatant is going to kill the target.
Yeah, I understand. Anybody else has any questions? I was also, it was really cool experiment, the one they're doing with the different individuals, the people actually taking the ETEC, because that will mean that their normal flora as they are might have different characteristics. Are there plans of looking beyond the species there, but for example, about diet, or what does it make that a person might have a different flora that would maybe make it more protective against certain strains?
I don't know. That would be in the hands of the collaborators in Bergen in that case. I mean, this is our part is to look at the competitiveness of the flora that is there. But of course, they are also interested in finding out if perhaps some of them have antibodies already against ETEC and how the immune system takes care of ETEC and what, I mean, so there are, of course, other differences that are possible than just competition, which is why also we don't worry too much about the fact that it doesn't correlate perfectly with how good inhibitors you have in your normal flora compared to how much symptoms you develop, because that's not expected. It's multifactorial.
Cool, very nice. Thank you so much, Sanna. If there are no more questions, anybody wants to say something? Otherwise, we're gonna go to the break and we're gonna have a 15-minute break. So we are back at 1:20, no, 20 past 1. We will be back with Olof's talk. Thank you again, Sanna, so much for presenting your work, and Petra's work, Dana's work, very, very extensive, I would say. And I wish you the best on the work moving forward. Thank you. See you all back in 15 minutes.
Bye-bye.